Hello and welcome back. In our last lecture we talked about atoms, the periodic table elements, and some of the properties of water. What we want to do in this particular lecture is go ahead and talk about carbon and some of the really cool things that carbon actually builds. Carbon atoms are very special in nature. They have 6 electrons, which if you look over here at its Bohr diagram, you can see that the first shell is filled, so there's 2 electrons in the first shield, and then in the second energy shield there are a total of 4 electrons. Now this makes carbon have the valence of 4, and it's also known as being tetravalent. Tetra is the root word for 4. And so having that 4 electrons in its outer shell gives it a special tendency to want to share electrons and bind with up to 4 other atoms sharing electrons. So if you look at nitrogen, it has a valence of 3, so it needs 3 electrons to complete its outer shell. And so it's very likely to join with other things. Oxygen has a valence of 2, and hydrogen has a valence of 1 because it only has 1 electron in its energy shield. So because of the properties of carbon, it wants to form covalent bonds. So that makes it a very, very special Lego of life, a very special building block. And so carbon is the backbone of all complex organic molecules. So if you look at proteins, DNA, RNA, hormones, sugars, lipids, vitamins, generally speaking, anything the human body uses or builds is going to have a backbone of carbon. Now, of course, carbon's not the backbone of oxygen. I mean, there's a couple of things that it's not the backbone of. It's not the backbone of water, but it is the backbone of all the really complex things that the cell has the ability to build. So it allows us to build simple things because of the tetravalent nature. It allows us to build simple things and very complex structures. That happens to be just a protein structure. So with carbon, we can build chains, we can build branching chains, so chains that have branches. We can build things with double bonds. We can build things that are ringed in structure. We can put rings together to form larger structures with rings. Now, carbon by itself, I like to think of is like a Swiss Army knife. So the base of the Swiss Army knife, so this actual part right here, I like to think of that as like the carbon, okay, the backbone of carbon. And if you want to think of a Leatherman tool, this is the backbone made by carbon. But you can see the Leatherman tool has, or the Swiss Army knife, it has different things that are attached to it. So scissors, knife, little saw blade there, which is kind of cool, a can opener. And so I like to think of these as functional groups. These are atoms. Well, we'll just consider them in the metaphor as being atoms. These are atoms that stuck to the carbon skeleton give the molecule special properties. So for example, in the Swiss Army knife, this particular functional group, the knife blade, gives you the ability to cut. This one gives you the ability to cut like scissors. This one gives you the ability to saw. So, it's not much different than what atoms do. So carbon being the backbone, there are functional groups that are actually attached to it. So the unique properties of organic molecules not only depend upon the carbon skeleton, but also the functional groups that you have attached to it. So just a couple of ideas about functional groups before we go into them. So functional groups give a molecule its reactive properties, so how it chemically is going to react and bond. It also is made of 2 or more atoms typically, and each functional group behaves the same from one molecule to another. So if we know how a functional group behaves, we can actually know how it's going to behave on each molecule that we actually study. And they're super, super, super important for living things. Let me give you an example of why they're important. So there's a difference between female lions and male lions. They have a different phenotype, a different appearance. They have different behaviors. They have different things they can do. Female lions can, give birth, and male lions can't. So, and one of the major differences between these animals is hormone, or their hormones, their sex hormones. So females have a lot more estradiol than they do testosterone. Males have more testosterone. Now you can see, generally speaking, those hormones look very similar to each other. Okay, there's a hydroxyl group here, a methyl group here. You can see that here and here. But you can see there's a couple of differences. There's an extra methyl group here and a ketone group here. This has a hydroxyl group here. So just by having a few different functional groups, this hormone causes this animal cells to do something different than this hormone does to cause this animal cells to do something different. These functional groups are very, very important. So I just picked out, I think, 4 functional groups for you to be familiar with for this class. The hydroxyl or alcohol group basically has a hydrogen atom bonded to an oxygen atom, which is then bound to the carbon skeleton. So you can have a carbon skeleton and there's the alcohol or hydroxyl group hanging off. And this is what it looks like. The stem means that it's attached to carbon. An organic molecule with this group is called an alcohol. So if you ever drink ethanol, you're actually drinking molecules that actually have hydroxyl groups hanging off of it. And so this is ethanol, and it's a liquid, and this is ethane, and it's a gas. Notice the difference between these. They have a very similar carbon core skeleton, but just because this has a hydroxyl group, it makes it an alcohol. It actually makes it a liquid. This has a hydrogen here, and it actually makes this have properties of being a gas. So functional groups are really super important. The next group is called the carboxyl group, and it consists of an oxygen atom double bonded to a carbon atom that is bound to a hydroxyl group. So this is how you would basically draw it. Carbon double bonded to an oxygen, singly bound to a hydroxyl group, and this is what we call the carboxyl group. This functional group can donate hydrogen ions, so this can actually donate hydrogen ions. Therefore, it acts like an acid because by definition, acids have higher and higher concentrations of hydrogen ions. So if you donate a hydrogen ion, you're going to be an acid. This functional group is contained in as a functional group on amino acids, which are the building blocks of proteins. I just wanted to show you this particular molecule called formic acid. This is the simplest molecule that has a carboxyl group attached to it. And formic acid is what you get to experience if you're ever stung by ants. So here's the stinger of the ant there, and the little venom gland is up in there. So when it stings you with formic acid, and your body senses when that chemical comes in, it senses it as pain. You have receptors that receive formic acid, send the information to your brain as pain. Now, if you ever taste an acid, okay, so an acid usually tastes sour. Okay, so hydrogen ions, when they're in large amounts in solutions, will taste sour. So when you eat, say, orange juice, it's kind of sourish, and because it has citric acid in it. So the amino group is the next group, and it contains a nitrogen atom that's bonded to 2 hydrogen atoms and then to the carbon skeleton. And it looks like this. You'd have the carbon skeleton, and then the nitrogen would be attached to that. So nitrogen and 2 hydrogen atoms. This functional group is found in amino acids, and it has the ability to accept hydrogen ions, so it can actually take and absorb hydrogen ions and attach it to it, and so it acts like a base. This is a very important a functional group in proteins because they have the ability to buffer. So we can, proteins can absorb hydrogen ions, and proteins will also have the ability to donate hydrogen ions. And this buffering system is found all throughout your body, but especially in blood cells. And this is just an amino acid just showing you what one looks like. So an amino acid has an amino group, and it has a carboxylic acid or carboxyl group. So amino, and since this is carboxylic acid, it's acid. So amino acid is what that means. And that's the simplest amino acid called glycine. The last functional group is called phosphate group, and it consists of a phosphorus atom double bonded to an oxygen atom and 3 other oxygen atoms to the carbon skeleton. So this is a very important functional group in making high energy compounds like ATP. ATP is cell energy. I like to think of it as a rechargeable battery that the cell uses, and it's released from the breakdown of food. This is an important building block also of DNA and RNA, and it contributes to the negative charge to many molecules, especially DNA. And it's an important component of cell signaling, of the cell signaling molecule called cyclic AMP. This is a chemical that we use to signal when some cellular events should occur. And this is what it looks like. Okay, so from those functional groups and from those carbon skeletons, we can build larger materials called macromolecules. And when you build macromolecules, new properties emerge from the union of those atoms. So macromolecules are polymers. So most macromolecules are polymers, which means they're long molecules made up of repeating units called monomers. Now, if you learn your root words, poly means many, mono means one. So that helps you to see how those things are built. Now, polymers are assembled by joining monomers together in a process called dehydration reaction. Some books will call it dehydration synthesis, but basically you're dehydrating or pulling water out between monomers, and it sticks them together, as I'll show you in a graphic in just a second. Polymers are disassembled by adding water, and this is called hydrolysis. Hydro means water, lysis means to break. So we can break polymers down by adding water. And if you ate something right before watching this lecture, you're doing it right now in your intestines. You have enzymes that are adding water to break down your polymers into monomers, and the monomers are being absorbed across the intestine of your across the wall of your small intestine. Many types of macromolecules with unique properties can be made from very simple monomers. So from simple building blocks, we can make complex things. And this shows you how we can take a monomer and build a polymer. So here you have a little short polymer, and at the end of it there's a hydrogen atom sticking out. Here you have a monomer that's not linked yet, but it has a hydroxyl group hanging off. Well, if I take and have an enzyme that can draw that water out, you have to have enzymes to do this. These are little proteins that help us to speed up these chemical reactions. But if I draw that water out, then I can actually stick these things together with a covalent bond. So now I'm making a longer polymer from adding monomers in. Now, if I want to take that long polymer and break it apart, I can take water plus an enzyme and break that bond right there. And now you can see the hydroxyl groups here, the hydrogen here, but I have individual monomers that are being released. Okay, so this one right up here is dehydration reaction, and this down here is hydrolysis. Now we have four major types of macromolecules that I'll talk about, and the first type is the carbohydrates. This is a term that includes both sugars and all of their polymers. So the simplest sugars or simplest carbohydrates are the monosaccharides. So mono means one, saccharide means sugar. So these are one monomer sugar units. They're sweet to the taste. They in water will form rings. Typically they're going to have a linear structure, but if you add them to water, 1, 2, 3, 4, 5, 6, most of them have 6. So when you add them to water, they actually form a ring structure. Okay, and that's a monosaccharide for you. So here's your linear structure right here. Add it to water, it forms a ring, and this one is a really important one. This is glucose, which is one of our main monosaccharides that we use. So they're very important fuel sources for cells. The carbon skeletons from that structure can be used to make other things. So we can disassemble the sugar, unwind it from its ring structure, and build amino acids and also fatty acids from it. So we can use it as a building structure or building block. Also, monosaccharides can be used to build disaccharides and polysaccharides. Disaccharides are sugars with 2 monomers. Polysaccharides have many monomers put together to build the structure. So because these sugars that I'm going to talk about have 6 carbons, we call them hexoses. Hex means 6, and then the root OSE means sugar. These are sugars with 6 carbons in them. So glucose is a very, very important fuel source. That's all your brain runs on. Galactose is an important sugar that you might find in something like milk. And then fructose is found in just about everything we eat. It's added as a component. Remember, high fructose corn syrup. And this is also found in table sugar, but it's a type of sugar that plants store their energy in. And then we eat them and break it down for our own energy. Disaccharides are formed by dehydration reaction. So water's lost between the monomers and we stick them together. So basically it's made of two monomers, therefore disaccharide. So here we have the two monomers. Here we have glucose here and here. We draw water out. Now remember, you have to have an enzyme to do that. And what we form from that would be a different sugar called maltose. This sugar has different properties than the two glucose molecules, so new emergent properties result from that. Now remember that suffix right there means sugar. So if we took glucose and fructose and stuck those together, we can form sucrose, and glucose and galactose will form the milk sugar called lactose. Important disaccharides for our purposes will be maltose. This is glucose and glucose, and this is an ingredient that we used in brewing beer. It's the sugar we feed the yeast in order to actually cause ethanol to be formed. Sucrose is going to be glucose plus fructose, and this is a table sugar, and it's a transport sugar in plants, but we eat it as table sugar. If you ever eat syrup or table sugar or sweetened with those kinds of things, you're eating sucrose. And then lactose is made from the combining of glucose and galactose, and this is the milk sugar that you enjoy when you drink milk. Now, some people have a problem drinking milk. They have what we call lactose intolerance, and this is where an enzyme called lactase is not working. Okay, a lot of people when they get older, that lactase enzyme doesn't work, and so you can only drink milk as a child, and then you get lactose intolerance later on. There are certain groups of people, depending upon your ancestry, that might have lactose intolerance, such as Asians and Africans. Europeans typically are going to be lactose tolerant because milk was an important agricultural product, and through thousands and thousands of years of evolving in Europe and having to drink milk in order to get vitamin C and vitamin D, those folks have mutations that allow them to be lactose tolerant. So I can drink milk and I enjoy milk products because of my ancestry. My ancestors came from Great Britain, and most of them, and then they came, or Scotland, and then they came from France and Germany. So I have the ability to digest those particular materials. Now, if you have lactose intolerance, when you eat lactose, it's going to cause gas to occur. This is because bacteria in your intestines have the ability to digest the lactose, and therefore they produce gases in the fermentation of that particular thing. Now, if you have the enzymes to digest lactose, they don't get an opportunity to eat that much of it, therefore they don't produce that much gas. So when the bacteria cause the fermentation of that causes the gas. But when you have lactose just sitting around your intestines, it actually draws water or pulls water out of your body and puts it into the intestines, causing you to have diarrhea, which is very unpleasant. Diarrhea, rhea means to flow or gush. That's the root of that particular word. Now, if you do have lactose intolerance, you can eat things like yogurt, because bacteria are used to pre-digest the lactose. So the yogurt doesn't have a lot of lactose, and the bacteria have the enzyme, they digest the lactose, and in the process of that fermentation, they create an acid called lactic acid, and that lactic acid, L-A-C-T-I-C, yeah, lactic. They produce the lactic acid, which ferments, which actually takes the milk, and that drop in pH, because they're producing acid, causes the milk to congeal to form that yogurt that you actually eat. So that's a pre-digested food that you're eating when you eat yogurt. Now, poly means many, and saccharide means sugar. So many sugars put together make a polysaccharide. So these are polymers of glucose. And these serve as a storage material, that we use to store energy. And glycogen is the storage polysaccharide found in animals. And here's a picture of it down here below of what it would look like. Each of those little parts is a glucose molecule, and you can see there's lots and lots of them. We typically store our glycogen in organs such as the liver and muscle. And when we have sugar like glucose, and when we eat it in excessive amounts, we can make lots of glycogen to a point, and then we don't use it any longer. And when we are exercising, the liver will actually release the stored glucose into the bloodstream, and our muscle will actually use it. At rest, our muscles also store glycogen, and then when they need to use it during physical activity, they release glucose molecules that will be used to make ATP in what we call cellular respiration. So lipids are hydrophobic. They consist mostly of hydrocarbons, which are carbon atoms with hydrogens attached to them. When you look at a lipid, this is what a lot of lipids are going to look like, just carbons and hydrogens. So I want to talk about different kinds of lipids. Our first kind of lipid would be the fatty acids. These are long chains of carbons with hydrogens atoms attached to them. At the end of the hydrocarbon, so at the end of the hydrocarbon making up a fatty acid, is a carboxyl group. Okay, that's also known as a carboxylic acid group. So a fatty acid has a carboxyl group, and then it's made of a hydrocarbon. And we have very important fatty acids that circulate around. We use them for building blocks, and also we use them for fuel. And there are certain kinds of fatty acids, like the omega-3 fatty acids and the omega-6 fatty acids that you eat in your food, and they're supposedly good for you. Some people actually think they're so good for you, they eat fish oil pills or lots of walnuts or different kinds of nuts to get these essential fatty acids. Breast milk has essential fatty acids that are used to build good brains for the infants that are breastfeeding. Now you can have a fatty acid that is saturated, which means that every carbon has a single bond between it. So lots and lots and lots of hydrogens are attached. And when at room temperature, these things will form a solid. Some fatty acids, though, have double bonds between them, and they form little kinks in the molecule, and they form oils at room temperature. So these are solid at room temperature, and these unsaturated fats and the saturated fats, excuse me, I had that backwards. The saturated fats are solid at room temperature, and the unsaturated fats are actually liquid at room temperature and form oils. So get that straight. Saturated means you have no double bonds. Unsaturated means you do have double bonds and form a liquid. And supposedly you're familiar with saturated fats being probably not as good for you and unsaturated fats being better for you. Don't think of fats as being bad, though, totally, because they're actually very beneficial to us. Glycerides are lipids that are made from the combining of a fatty acid to a glycerol, which is a little sugar molecule. And a monoglyceride is one fatty acid attached to glycerol. Diglycerides are two fatty acids, just using your root words, and then triglycerides, of course, are three fatty acids, and that's the most you can actually put on that glycerol molecule. We use these as an energy storage molecule. You can see that 1 gram of fat contains 2 times the amount of energy as 1 gram of sugar, so we do crave fat. Many people crave things like bacon, which is a very fatty type of meat. We use these glycerides also for insulation and for protection. So insulation, some animals have a very thick fat layer around them, humans not as much, and protection. So your eye is packed in fat so that it doesn't just float around in an eye socket. We also protect our internal organs with a layer of fat around them. And so this one right here would be a monoglyceride. So you can see it only has one fatty acid attached to the glycerol molecule. Here's your glycerol molecule. It's a 3-carbon sugar. And, because it has hydroxyl groups on it, it can draw water out and stick these things together. So there you just saw it stuck together. These happen to be an ester, what we call an ester linkage. I'm not going to ask you about that, but that's what that bond is called between those. So you know that these are 3 fatty acids attached to glycerol. So what kind of glyceride would this one be? And yeah, you would be right if you said triglyceride. Of course, this has less energy storage than this one. This has a lot more fatty acids attached, therefore much more energy that's stored in it. Phospholipids are another type of lipid that compose our cell membranes. I'm not going to make you, draw this structure. I will make you draw this structure. So a phospholipid has a hydrophilic or water-loving head, and it has two hydrophobic fatty acid tails that hang off of it. Sometimes they're saturated, sometimes they're unsaturated. This one you can see has a little kink in it, and that gives that just unique properties to that fatty acid. You have different kinds of components of your cell membrane and different kinds of fatty acids attached to the hydrophilic head. So you should be able to draw this structure knowing it has a hydrophilic or water-loving component and a hydrophobic component. Now, when you put these phospholipids into water, they actually aggregate in such a way that the hydrophobic tails shield themselves from water, and the hydrophilic heads will actually affiliate with water. And this forms a cell membrane. Okay, so we have a hydrophilic head, hydrophobic tails, hydrophilic head. So this would be water out here. This would be water inside the cell. So it forms a membrane around the cell. So your cell membrane looks like this if you were to look at the components making it up. Again, this portion is shielded from water. This portion is affiliated with water. So it forms a bilipid, a two-layer structure. Here's one layer and here's a second layer. So yeah, that's a really important part to know, because cell membranes make up all the trillions, 30 trillion or so cells that are inside the human body. So steroids are another type of lipid, and they consist of 4 fused rings with different functional groups attached to them. Cholesterol is the precursor to our steroid hormones, our steroids in our body, and the cholesterol is also an important component of cell membranes, making up our cell membranes. So important animal steroids or steroid derivatives would include testosterone, estrogen, vitamin D, okay, is a steroid derivative. Cortisol is a steroid hormone. It's a stress hormone. And then another steroid derivative would be the bile salts. Bile salts are actually stored in our gallbladder, and they're released when you eat fats, and they help to break down fats. And this is what cholesterol looks like. So you can see it's got 1, 2, 3, 4 fused rings, which makes it a steroid. And you can see it's got unique functional groups to it. These functional groups will differ or deviate depending upon if the thing is vitamin D, bile salt, testosterone, or estrogen. So from this backbone, you can build many different complex things. Many people think that eating cholesterol is bad for you, and that's pretty sad. You have to eat some cholesterol. Your body does make some cholesterol, but you do have to eat cholesterol in order to survive. Now, there are people that have hypercholesteremia, which means they produce too much cholesterol, and they may have to go on some kind of a statin, which is a cholesterol-lowering drug. You don't want too much cholesterol, but you don't want too little of it, so got to be careful with the cholesterol. Eicosanoids are another lipid that's made from arachidonic acid. This is a component of cell membranes, and it's something that you eat. And we can build from these arachidonic acid, we can build leukotrienes. Leukotrienes are lipids that are involved in coordinated responses to injury or disease. It's also involved in an allergic reaction. Some of the allergy medicines that you take knock out leukotriene production, thus knocking out allergic reaction. Prostaglandins are involved in the sensation of pain. These are natural chemicals we create from arachidonic acid, and they're involved in the sensation of pain. When you take something like an aspirin will stop the production of prostaglandins and thus stop your production of pain, because there's no more prostaglandins creating it. Prostaglandins also will trigger contractions of the uterus, initiating labor. So that's kind of cool, a cool chemical. Okay, so our next macromolecule will be the proteins, and proteins are chains of amino acids that are connected in very specific sequences. So an amino acid is a monomer of proteins, and its structure looks like this. And I do expect you to be able to draw the structure, so you might want to practice that, but essentially you have a carbon in the center, and that carbon is attached to a hydrogen and to an R group or side chain. These R groups or side chains are variable. There's 20 different kinds of them that make 20 different kinds of amino acid. We have on one end a carboxylic acid or a carboxyl group, and then we have on the other end we have an amino group. Okay, so we have an amino acid is what this is with the R group. There's 20 different naturally occurring amino acids in the humans. There's actually more than 20 amino acids, but not all of them are found in humans. 8 of them are essential to humans. That means in these amino acids, you have to actually eat them in order to have the building blocks to make the proteins. So 12 amino acids we can actually make from sugars or fatty acids or different components or other amino acids. But 8 of them you have to eat in your diet. So if you are a vegetarian, you want to be careful. You want to make sure you eat different kinds of varied vegetables in order to ensure that you get all the different amino acids. So, for example, if you went on an all rice diet, you would die. If you went on an all corn diet, you would die. If you went on an all bean diet, you would die. But if you mix them like rice and beans and rice and corn and beans and you mix these together, you get all the essential amino acids that you need. So in a survival situation, if you ever want to prepare for the next pandemic and have 2 weeks of stored food, rice and beans or corn and rice, all of these store very easily. They're cheap, and you will be able to survive getting all the amino acids you need. So in a survival situation, get you a multivitamin, get you some of these materials here. And then, of course, you want to have some carbohydrates and some oils. And for a very small amount of money, you can have pretty easy survival food to last a pretty long time. The CDC recommends you have 2 weeks of stored food at any given time, because if we ever had a pandemic of flu, that would maybe knock out the ability of you to go to the grocery store for up to 2 weeks. Add some masks to protect you from getting the airborne virus, and you're good to go. So amino acid properties are determined by side chains. So those R groups that we looked at just a second ago, sometimes those R groups are nonpolar. They don't associate with water. Sometimes they do. Sometimes they have acidic properties or basic properties. And this gives you each individual amino acid being unique. Now, in this class, I'm not going to ask you to memorize the amino acids. If you ever have a class like biochemistry or something of that nature, you will have to memorize these, but not for this class. I just put these out to show you what some nonpolar amino acids look like. So each of these are nonpolar. They don't affiliate with water. And then these over here are polar. They would affiliate with water. Some of them are acidic because they donate hydrogen ions, and some of them are basic because they have the ability to accept hydrogen ions. So different proteins have different properties because of the different arrangements of amino acids that are attached together making up that particular protein. Now, polypeptides, which mean many peptides or many amino acids, they're assembled by an organelle called a ribosome. And in our next unit, we'll talk about what ribosomes do and how they work. The order of the assembly of a protein is basically coded in a molecule called RNA. There is a sequence of RNA that codes for the production. They're like blueprints that code for the production of the protein. RNA goes to a ribosome, is read by a ribosome. The instructions are read, and the protein is actually composed. The sequence of RNA, though, is made from your DNA, so all the coded instructions to build anything in the human body comes from the coded instructions from DNA. Of course, we have different DNA and different mutations that create different proteins and give different people different kinds of properties. The amino acids are joined together in a very specific sequence to build proteins, and we attach those in what we call peptide bonds. Some proteins are composed of thousands and thousands of amino acids attached together in a particular sequence. Other proteins have smaller numbers of amino acids. So the composition and sequence of amino acids gives the protein its structure and therefore the function that it performs. So I do expect you to draw a peptide bond. I mean, it's pretty easy to do. So we have one amino acid over here. Now, because we have the hydroxyl group here, I'll show you what it does to the amino group here. So here we have our amino acid. We can draw water out and stick this amino acid to this amino acid in a peptide bond. Let me kind of show you what that peptide bond looks like. It basically would have a line coming across. Okay, so that's a peptide bond right there that you create because you drew water out between that hydroxyl group that was over here and a little hydrogen that was over here. We drew that out, and that sticks these together covalently, and we do have to have enzymes to do that. So always remember, enzymes are important to do those things. Okay, well, the structural levels of proteins can exist as a primary structure, and this is the linear unique sequence of the amino acids. If you change the order of the amino acids, you change the protein. So that's what primary structure looks like. It's just a sequence of amino acids. Okay, but proteins are more than just a primary level of structure. Proteins also have secondary structure, and this is where the protein, because of the amino acids that are in them, they can actually form folds and coils. And it's because of hydrogen bonding between adjacent amino acids. And so we can form coils or tubes, and we can form sheets. This is called beta-pleated sheets, and this is called an alpha helices or an alpha helix. Okay, so a protein can take on some unique shapes. It can have folded portions and it can have tubular portions based on secondary structure and because of that hydrogen bonding. Now, if I boil a protein, I break the hydrogen binding, and will damage the protein, maybe even irreversibly. Tertiary structure is the irregular contortions of a protein due to all kinds of interactions. So if you have amino acids that are hydrophobic, they'll actually cause the protein to coil so that it will shield that particular portion of the amino acids from water. Sometimes we have hydrogen bonds that will form little kinks and coils. Sometimes we have another functional group called sulfhydryl groups that will form a disulfide bridge between adjacent amino acids, causing a big kink to form. And then you can have ionic bonds. There's all kinds of little interactions that you can have that coil a protein into its tertiary structure. So you can take a linear protein and you can take and bend it and coil it and make it have a unique three-dimensional structure that gives it its function. Some proteins, but not all, but some proteins will be made of two or more polypeptides joined together, and they'll have quaternary structure. So there's a couple of unique proteins in in animals. Collagen, this is what collagen looks like. It's actually 3 twisted proteins that give the final protein great strength. Collagen is found in your skin and hair. It's it's the most important, most abundant protein in the human body. Here you can see in hemoglobin there are 4 different, 4 different polypeptides that come together to form the final molecule we call hemoglobin, and this particular molecule carries oxygen in your blood cells. So this is just a graphic showing you primary structure, just a linear sequence of amino acids. Secondary structure showing you how it can form beta-pleated sheets and alpha helices because of hydrogen bonds. All those other bonds and interactions will form a 3-dimensional final structure of a protein, and then if you have more than one protein added together, you can have quaternary structure. Now these structures can be denatured. This is where they unravel, so you can actually destroy the secondary, tertiary, and quaternary structures of a protein by boiling it, exposing it to changes in pH, and exposing the protein to various kinds of chemicals like alcohol. So when we denature proteins, a lot of times that's irreversible, especially if you heat it or expose it to certain kinds of pH and cook it with pH changes. So, you don't want to cook your hand on a stove. You do want to cook your food to denature the proteins, because if there's any microorganism living in your food, you want to make sure you cook it and kill it. We do pickle things by changing pHs or using acids, and so various kinds of chemicals we use to clean with, like alcohols and bleaches, because it will denature proteins and kill them. Now notice that the primary structure is not denatured. That's the linear sequence of amino acids, and you can't denature that. You have to actually chemically break that down using enzymes to break the bonds between them. And this is a normal protein, and if I put it in a refrigerator, I could denature it, or a freezer, I could denature it, and then when I bring it out, I can actually renature it into a normal protein. There is the ability, though, to denature in finality a protein by doing certain things to it, like cooking it. And our last of the macromolecules are the nucleic acids, and these are made from monomers called nucleotides. Nucleotides have three parts that they use to build the building block. They have a phosphate group, a pentose sugar, and a nitrogenous base. And this is what a nucleotide looks like here. This is what the actual polymer looks like over here. You can see it has a lot of nucleotides attached together that form the molecule. And so DNA and RNA, these are molecules that are going to be made of polynucleotides. So the nucleotide is made of a phosphate group. We saw that as a functional group. It's made of a nitrogenous base. The nitrogenous bases can come in one of 5 flavors, depending upon if it's RNA or DNA. So, typically you have cytosine, thymine. Thymine is found only in DNA. Uracil is only found in RNA. Or you can have double-ringed nitrogenous bases, adenine and guanine, and these would make up all of the different kinds of nitrogenous bases. So not a lot to select from. And then it has also a pentose sugar, a 5-carbon sugar. Penta means 5, ose means sugar. So in RNA we have ribose. So ribonucleic acid is what RNA stands for. And in DNA we have deoxyribose. Deoxyribonucleic acid is what DNA stands for. So these would be the building blocks of your nucleic acids. Now, a gene is a specific sequence of the A's, T's, C's, and G's, the nitrogenous bases. So, for example, that would be a unique message, coded instruction, that we use to make different RNA molecules, which then eventually make different proteins. So different codes make different proteins. So you have a library of information inside of your cells locked in the DNA molecule. DNA is a template that we use to make an RNA molecule. RNA can actually leave the cell, as you can see going through this little nuclear pore here, and it goes to a ribosome. And the ribosome is like a reader of the RNA, and it actually reads the code instructions to make a protein. And that protein will go off and do a different job. Maybe it becomes collagen, maybe it becomes hemoglobin or an antibody, or it does something different. Okay, and we make lots and lots of different kinds of proteins, maybe somewhere in the order of maybe 90,000 unique kinds of proteins from the 20 or so thousand genes that are inside of our DNA. Okay, well that completes the chemistry video number 2. And in our next lecture, we're going to begin to talk about cells and enzymes. So until next time, I wish you a good day. Make sure you're doing all the things that you know you need to do to be a hard-working student. Make sure you work on study guides. Make sure you work on root words. Study hard. Watch portions of this lecture. Make sure you do your reading. And growth only comes from great effort. So if you want to grow and have a growth mindset for this class, you're going to have to work really hard. So I wish you all the luck with that. If there's anything I can do to help you, make sure you get in contact with me. So until next time, I wish you a good evening or a good morning whenever you're watching this lecture.